Dr. Sage here, continuing our discussion on microbial genetics. In particular, today we're going to discuss DNA recombination events. By the end of this video, you should be able to explain the defining characteristics of a recombinant organism and describe 3 forms of horizontal gene transfer used in bacteria. Okay, so what is recombination? Recombination is an event in which one bacteria donates DNA to another bacteria. The end result is a strain different from both the donor and the recipient strain. Some more definitions. Plasmids are extrachromosomal DNA adept at moving between cells, and recombinant is any organism that contains and expresses genes that originated in another organism. And what is horizontal gene transfer? It's any transfer of DNA that results in organisms acquiring new genes that did not directly come from parent organisms. So some more details about plasmids. They're small circular pieces of DNA that can replicate independently of the bacterial chromosome. They allow for transfer of DNA between cells. They're found in most bacteria and in some fungi. They contain at most a few dozen genes, and they're not necessary for survival, but they often carry useful traits. 3 examples of horizontal gene transfer are transformation, transduction, and conjugation. We'll learn about each of them independently. First, conjugation can also be thought of as bacterial sex. It's a mode of genetic exchange in which a plasmid or other genetic material is transferred from a donor to a recipient cell via a direct connection. It can occur in both gram-positive and gram-negative cells. Okay, some more details. Let's say we have this cell that's called an F+ donor cell. The reason it's called an F+ donor cell is because it contains the F plasmid. F stands for fertility. The F plasmid will allow creation of a pilus or sex pilus, which is a tube joining 2 bacteria cells. So the pilus of the donor cell attaches to the recipient cell. The pilus contracts, drawing the cells together to make contact with one another. Then one strand of the F plasmid DNA transfers from the donor cell to the recipient cell. The donor then synthesizes the complementary strand to restore the plasmid, and the recipient synthesizes the complementary strand to then become an F+ cell, which can create its own pilus now. So what's happening by the transfer of this F plasmid is it's turning this F- recipient cell into an F+ donor cell, because in the end, both cells now have the F plasmid. Conjugation can also transfer resistant plasmids. So resistance plasmids carry genes for resistance to antibiotics or other drugs. They're commonly shared among bacteria through conjugation. It can confer multiple resistances to antibiotics. R factors can also carry genetic codes for resistance to heavy metals or for synthesizing virulence factors. So this is one way that there's a buildup of antibiotic resistance bacteria, is that bacteria has an R plasmid that contains resistance to some antibiotic. They then transfer that antibiotic resistance plasmid to another bacteria, converting them to also be now antibiotic resistant. Another type of horizontal gene transfer is transformation, which is capturing DNA from solution. Bacteria, if there's DNA floating around outside the cell, they can actually bring that DNA into their cell, and they can even integrate it into their chromosome. So transformation is the acceptance by a bacteria cell of small fragments of soluble DNA from the surrounding environment. Competent cells are cells that are capable of accepting genetic material through transformation. How do we discover or learn about transformation? Well, it was discovered through an elegant experiment conducted in the 1920s by Frederick Griffith. He worked with encapsulated and non-encapsulated Streptococcus pneumonia and laboratory mice. Encapsulated strains have a smooth colony appearance. Strains lacking in a capsule have a rough appearance. Okay, so this is essentially what Griffith did. He took the rough strain, which is non-virulent, and injected into the mice. Now, being non-virulent, the mouse lived completely fine. He then took a smooth strain, which a smooth strain is virulent, so it would kill the mice if it was injected into the mice. But in this case, he heat killed the smooth strain. In other words, these bacteria were now dead. Okay, so even though they're injected into the mouse still lives. He then took the non-virulent rough strain and the heat-killed smooth strain, which either of them independently, if you inject the mouse lives. He injected them both at the same time, and what happened was the mouse died. He then recovered from these dead mice a smooth strain that was living, injected into the mouse, and again the mouse died. So what was happening was the rough strain, the non-virulent strain, was taking up DNA from the dead smooth strains. So the bacteria were dead, however they still had DNA. That DNA was released into the environment. That DNA then got taken up by the rough strain, and that converted the rough strain into a smooth strain. And that's why the mouse died. So this was the first evidence of transformation among bacteria. Another type of horizontal gene transfer is called transduction. Transduction is a process by which a bacteriophage serves as a carrier of DNA from a donor cell to a recipient cell. All right, first thing to note, bacteriophage. Phage means virus, bacterial means bacteria. So this is a virus that infects bacteria. Transduction occurs naturally in a broad spectrum of bacteria, and the participating bacteria in a single transduction event must be the same species. All right, so what this means is when a bacteriophage infects a bacteria, it injects its DNA into the bacteria. That DNA can then be integrated into the bacterial chromosome. So you can have generalized transduction, where random fragments of disintegrating host DNA are taken up by a phage during assembly, and any gene from the bacteria can be transmitted. Or specialized transduction, as a highly specific part of the host genome is regularly incorporated into the virus, explained by the prior existence of a temperate prophage inserted into a fixed site on the bacterial chromosome. The prophage DNA separates from the chromosome, carrying host genes with it. Something else we can have are called transposable elements. So transposable elements, also called transposons, are called jumping genes because they shift from one part of the genome to another. They were first proposed by Barbara McClintock in 1951, and jumping genes are widespread among cells and viruses. So here's a transposon, or a transposable element. Okay, it's a chunk of DNA where you have the transposase gene that codes for an enzyme called transposase. And then surrounding that transposase gene are some inverted repeat sequences of DNA that transposase enzyme will recognize. So the transposase then facilitates recombination between inverted repeats. The transposon is cut from its original location and then inserted into a new location. So what this means is this piece of DNA has the ability to cut itself out from the chromosome and hop or reinsert itself somewhere else inside the chromosome. Types of transposable elements: we have insertion elements. These are the smallest transposable elements and consist of only 2 tandem repeats. Or a retrotransposon, a type of transposable element that can transcribe DNA into RNA and then back into DNA for insertion in a new location. Other transposable elements contain genes that code for antibiotic resistance or toxin production. So what are the effects of having transposable elements? Well, general effects is they can scramble the genetic language because they're inserting themselves into different places in the genome. They can be beneficial or adverse depending upon where the insertion occurs in the chromosome, what type of genes are relocated, and what type of cell is involved. Now, in particular in bacteria, transposable elements can cause changes in colony, morphology, pigmentation, and antigenic characteristics. It can involve replacement of damaged DNA, and it can transfer of drug resistance between bacteria. Pathogenicity islands have the ability to make their host pathogenic. They contain multiple genes that are coordinated to create a new trait on the bacteria. Islands flanked by sequences that look like genes for transposable element enzymes. Okay, well this was your introduction to DNA recombination events that occur in microorganisms. Until next time, this has been Dr. Sage.